So I begin with a little bit of a background or a view on machine learning. Okay, so prediction is the acid test for understanding. What it means is that, remember, when we looked at what are the preferences for particular residues or particular amino acids to be involved in protein interactions, we saw that some types of amino acids are more often seen in transient protein interactions, right? Now, you may assume that this relates to the fact that the biophysics is such that you see these kind of residues more because, for instance, when you look at DNA, you will see more positively charged residues because DNA is negatively charged, right? There's a simple interaction, and by seeing more positively charged residues, you can guess that the binding partner is something negatively charged, right? That is sort of understanding. So there, as long as it's statistically significant, you assume that some science is implied. But that ultimately is correlation. It's not quite understanding. It could be that this correlation actually has nothing to do directly with the interaction. It could be an effect of something else. It could be the effect of, I like this example of when you look at car color. So when you look at the color of a car that has the fewest number in a garage, in a workshop, then the answer, what is the color? The answer is white, right? Why is that? Has nothing to do with white cars being in any way more stable or anything like that. The point is they do fewer accidents because white cars are easier to see, except for in places where there's constant snow. But on average, for actually including Germany, so the statistics are very clear. White cars are relatively in fewer accidents than any other color. So there we have a correlation. So it has nothing to do. There's no causation between color and fitness of a car. Fitness of the engine has nothing to do with the color. It's not that the color goes into the engine and the white color destroys the engine less than the red one. The red is the killer or whatever. Now the question really is, where is understanding? If I can predict, do I understand? Well, clearly the other way around is true, right? If I cannot predict, then I don't understand. Whether there is a statistical correlation between positively charged residues, if I use that and predict DNA binding, and if most of my predictions do not bind DNA, then statistics and science do not correspond to each other. I clearly have to be able to do prediction. If I can prediction, then I assume that somewhere underneath making this prediction is understanding, right, in the model. Now, what is the story here if the model is machine learned? Do I still have understanding? And what I'm telling you is a perspective. It's my view, let's call it like that. And the view starts with this idea of black magic. And the view starts with this statement that there is something in this machine that in fact learns maybe some truth, learns some rules, but it's in the machine, it doesn't get out. Let's begin with the first part that is essential in machine learning. You have to do the cross-validation. So you take some of the data, you take some other part of the data that you hide under the table, and you set up the machine. The next story is the part that you hide under the table. You have to use everything in your database exactly, in your data set exactly once. Then you have to sort of do a cross-validation, meaning here exactly once is this one here. And then in fact this is not enough. You have to in fact do a threefold cross-validation where you essentially do all optimization on the cross-training. All optimization includes you're choosing an SVM over a neural network. You're choosing 50 input units over 49. All of this is decided on the cross-training. The testing set is used exactly once. When all the development is done, you know this is my best method. You're not comparing two methods. There's one method. And the only reason why you have the test set is actually to estimate performance. This is the data set. All you want to do with this one here is tell the user, if you apply that tomorrow to a data set that I have not seen before, what do you expect? Is the performance. That's what the test set is for. All comparisons of method development of anything I do are on the cross-training level, okay? Now, this is back to the story that Jajun brought up on Thursday, and some people were stumbling here. So you have to really do the rotation exactly once. this somehow the way he showed it in his in this figure was not the same as this one. This is, remember that you have to sort of add up these 4 pieces here. That's one cross-training set. These 4 pieces, one test set. Right? Everybody remembers that. Now, this is not enough. You cannot just randomly split the data set. You have to watch that there is no overlap in the data set. For instance, if you had a family of proteins here, you cannot use one for training and one for testing or cross-training. So you have to group them in a way in which the training and testing will be done from different parts. Thank you for appreciating my smile. This is the kind of thing that takes a lot of work, and in the end nobody recognizes it. I don't know, not many people have ever smiled about my smiling. I'm not really an artist. Anyway, I know that. But now finally somebody sees something in my art and I'm happy. Thank you. So this is still not enough. You need to do something that we refer to as what I typically refer to as the pre-release data set. So this is when you develop your method, you took a few months to do the machine learning, a few months to do all of that. In these few months there's new data, hopefully, that you can use to do a final check, an additional check. That's one way of doing it. Another way of doing it is this example here for the hotspots, where the method really only learned to predict protein interaction interface sites and could predict hotspots without having trained it. Right? This is another example. There we clearly know that the method understood something from the problem underneath. There is not a problem with the cross-training because it was not used for training. Right? But that's a rare case. And then when you do that for many proteins, you see that it works. Here's the story about error estimates, and we can get over that. Now, we've done all of this cross-validation right. Now the question is, do we understand? And that brings us to this issue. The black box is this machine that contains the information in there. Maybe the machine understands, but we don't. The typical idea of a black box is that if there were some understanding in the machine learning device, it's hidden in the box. The box is black. If there is anything in there, I cannot see that. Is that true? So this is I would call the common misunderstanding. And this is the common misunderstanding. The word common implies that most people have that. In fact, most people who are experts in the field share this misunderstanding. But it's a misunderstanding that comes from the following issue. So first of all, it is the assumption that we cannot open the box and look inside what the rules are. Yes, actually we can do that. Some of you were in the last semester lecture where the secondary structure prediction was a topic. So in these original neural networks that predict secondary structure, you can really see what are the patterns in the input that imply secondary structure. So you could see these typical rules that people had. So in fact, I sort of opened the box and I could recover every known rule. So when people have sort of biophysical rule, proline breaks the helix is one of those very simple things. And a couple of these rules, and all of these rules, the neural network had learned. Okay? But now you could take these rules, put it into random forest or some other rule-based system, and you saw that using these rules alone, prediction performance was 58% or something like that. The neural network had 62, so it was better. That means it had these rules, but these rules alone were not good enough. There was something on top. There were more complex rules. When you got those out, you saw that if at position, there was a neural network somewhere here, I can just use that. So if there is a K at position 12, and then there is an F at position 4, and then there is an A next to the center P, then the probability is that it is. And the rules get very, very complex. And nobody who is an expert in the field understands anything about what do you mean if a K is at position 14. So there's a neighborhood of Ks that people understand. But the moment it gets to the level of complexity that the neural network actually had, these rules could not be translated to something that mapped to our brain. right? And ultimately, the problem here is not that we cannot get the rules out. The problem is that we apply machine learning devices to problems that are so complex, that there are no simple rules. So when I have a simple problem, a problem where simple rules explain it, I don't need a machine learning device. I only need machine learning devices in situations where I actually don't have that rule, okay? Maybe there will be a theory that spans underneath, and one day I will be able to do that. Maybe if we looked at all the data from the stars, we could machine learn the shape of the solar system without understanding really how the solar system works. But I would actually challenge that at the moment you have a complete machine learning map of it, you will see exactly the same system come out, and maybe ultimately the equations will bump at you. But that, at this point, is a speculation. Ultimately, the most important thing here is machine learning is typically applied for situations or problems that are so complex that when we get these rules out of the black box, they are there. The box is not black. They are in there, but we don't understand them, okay? And that is a big misunderstanding of most people. It's not that we cannot get the rules out, we can. And so one simple way you sort of can look at different strength of predictions, you can look at the impact of different features, but ultimately the problem is we cannot write these rules in a simple way. Okay? Because machine learning ultimately is applied to problems that are so complex that we don't understand the rules. Now, when we look at the protein-protein interaction story, some of these initial stories that are complicated get even more complicated. Again, feedback, so we want to predict now that two proteins bind that are in fact in contact. That means physical interactions. So green binds to the blue here, for instance. Green binds to the ochre here. And these interactions are physical and they're transient, meaning at some point of the life of the protein it binds, at some other point of its life it doesn't. And what we want to do is we want to sort of predict it through some sequence-derived features. For instance, one idea would be I take 9 consecutive residues from one protein, 9 consecutive residues from another protein, and I try to predict the region in which it's in binding. That in itself, and then I sort of slide the protein through. I told you that one in itself it doesn't work. There are all kinds of things we have to do in order to make that work. We have to predict the surface, we have to predict external interfaces in binding sites. This is what we have been doing over the last weeks. We have to look at internal residues. That's what we briefly talked about last semester. We have to predict subcellular localization. We talked about that one. We have to work on domains and improve alignments. We talked about that last semester. Then we can sort of put all of that together, and Tobias did that. But before we now can combine it, actually we have to prepare the data a little bit more. And the first issue here is the problem of redundancy reduction. So in a usual case where we classify protein A and protein B, one used for training, one used for testing, to predict, for instance, subcellular localization, we have simple circles. For the protein interaction story, now we are not having simple circles because now we're actually not having A to cluster, we have to cluster the interaction AB. So each one of my circles now is actually a pair of proteins, right? And that means that the story, the simple story, suddenly gets more complicated. Let's begin with the simplest way of looking at it that was presented by Jean-Guy Park and Edward Marcotte. So they distinguished 3 different cases, where case 1 is such that I train on A interacting with B. And my test set, or my test set contains AC and BD. Okay? My test set does not contain AB, but it contains the protein A, and it contains the protein B, pairing to proteins that are not these two. Okay? That's case 1. In case 2, one of them contains this, and let's just look at the 2 extremes here. Case 3, none of these 2 is ever used for testing when any of them is used for training. A binds B is part of my training, then I cannot test on A or B in any configuration or any other protein, okay? That's the case 3. Now, most publications until that point, which is in 2012, had in fact constructed data sets that are completely mixed up these cases. And then what they observed is that in fact for this case, so the pair is not in training, but each protein is in training, performance appears to be higher. Okay? In terms of curve here, recall versus precision curve, 2 methods, pipe and sigprot, both methods predict from sequence alone whether or not a protein pair AB binds. Okay? You see that the levels of accuracy that you can reach for some pairs appear to be very high, but mostly they're relatively low. Arguably, the most interesting region for proteins is somewhere here. So you want to have a good prediction for a few proteins. You want to discover some new interactions, and you don't want to look at too many false positives. So what really is most important about this ROC curve here is the beginning. Anyway, the important part here is, in red you have C1, so A or B have been used in training. So the A and B both are also in testing. There's some overlap, again, not of the interaction. And C3, none of them has been used. And you see that green is much below red. The same is true for this method. Okay? So it makes a difference. Now comes the next issue. The next issue gets back to the data. On the level of the data. In the data, what we have is we have the protein, the interaction between AB, which is experimentally measured. Okay? And I'm going to have many of those interactions, and I'm going to learn to predict that A and B bind. Okay? Some thousand. So we're going to, by the way, another thing that we realized is we really have to do that, at least in our hands, we have to do that on a particular organism. So I cannot develop, it's the first tool that I've developed where we have to be organism specific. So for instance, we began with human. So we're going to look at all the human proteins for which we have interactions, and we have several thousand pairs for which we have sort of good interaction. For machine learning, that's not enough. What else do we need? There are a few thousand pairs of proteins AB that bind, that are experimentally known to bind. What do I need on top to do machine learning? My question. Yeah. That's true. We need a redundancy reduction, and that is the issue that I talked about before. But I cannot do machine learning only on the proteins that I now bind. We need pairs CD where we know that C doesn't bind D. Exactly. What's the problem with that? What, clear? Yeah?
Maybe much less people do experiments on protein that not bind.
Why?
So you don't know.
Why? Why would fewer people do experiments that proteins don't bind? Actually, I would, so I'm not entirely sure how much you know of experimental biology, but also you all have programmed. I know that most of my ideas I had were wrong. Most of the things I tried went wrong. So most things fail. Most likely, in fact, most people spend most of their time trying to show interactions and they fail. Meaning that mostly they show that things don't work, right? Why don't you see that in the literature?
Because then it would never get through a peer review?
Right. Because the fact that I don't manage to make it bind doesn't mean that it doesn't bind, right? Maybe I didn't find the right conditions. I will not publish that. So it's one thing, if I can show that A binds B, I'm going to make sure that I really find this binding, and I look at different ways of looking at it, and I know how to do that. But showing that it doesn't bind means I have to do more experiments. I need to find one case where they bind, or I have to be able to show that it can be repeated, the experiment. But I need a publication is A binds B. In order to say that C doesn't bind D, I need to make many more experiments. I need to try many more conditions. All I need is one condition that works, and that I show, and that's enough. For showing that it doesn't work, I have to sample through all possible conditions, right? And that is highly non-trivial experimentally. And if I do begin to do that, then the danger is that tomorrow somebody finds a condition that I didn't try, and on top shows that I was wrong. So I don't gain anything from it. I can only lose. From this, well, somebody can show that I'm wrong, okay, but this is science, right? This can always happen. But putting up positive evidence is much easier, and it's therefore, because it's easier, also less likely that I'm going to be falsified on that. This is highly dangerous. So it's not encouraged because it is not easy, it's not encouraged because it is dangerous, and it's not encouraged because it's more difficult to get it through peer review, because partially all of these reasons and others, right? So that means, where do we get the negatives from? We could do machine learning on only positives, but so far nobody has really succeeded in showing that this works better than the tools that we have out there. Maybe there's a solution out there, and so for the topic that we will begin on Thursday, namely the effect of sequence variation upon protein function, is the same story. We have a lot of positive cases, and people are trying to make it work, and this is another high incentive to make it work, but we haven't made it work so far. Anyway, so where do we get the negative data from? Where could you get it from? Some of it is experimental, but very little. So Dimitri Fishman, for instance, has collected an experimental data set of proteins that are known not to bind. Some of these data sets exist, but they are small. So what do we know? By the way, there is an issue here. When we predict protein-protein interactions, this is a similar story, but it's somehow not as severe. When I predict binding, so I predict that a particular residue in a protein binds to DNA. Okay? Maybe the random chance in the protein 1 in 20 of the residues bind DNA. So random would be 1 over 20. Now I'm going to run my method here in human. So I'm going to look whether or not A binds B. A is a human protein, B is a human protein. What is the random background? And the random background depends very crucially on how many proteins does A bind. So if every human protein interacted with 1,000 proteins, or let's say 2,000, which is easier, then random would be 10%. Do you believe this is the case? Is 2,000 a good number, do you believe? So let's just put it out there. We do not know what the right number is. If, again, what we assume is that when we look at the final plot of degree, so number of interaction partners that a protein has, and here the distribution, we assume some zip. So 1 over x or something like that of the type 1 over ex. So of the type that we have very few hubs, many proteins have very few binding partners, some have a lot. Okay, we have a curve like that for the data today, but today's data is somewhere. It is also a zip. It's much lower. What we do not know is how much higher the real curve will be. And that question, how much will we lift from here, is ultimately the way to answer the question, what's the average number of interaction partners that a protein has? We have actually no clue. So we absolutely know that we are very confident that the average will be higher than 2. And we are very confident that the average will be lower than 20,000. So protein, not every protein interacts with every protein. That's also very clear, right? And I would argue that 2,000 is a high number. But whether we have 1 positive in 100 negatives, or it's 1 in 10, or it's 1 in 500, or let's say 1,000, these 3 all have publications going for them. So for all 3 numbers, and you see we have 3 orders, 3 orders of magnitude, right? So the lack of knowledge is we're not sure whether it's 1 in 10 or 1 in 1,000, which is a dramatic lack of knowledge, right? People begin to feel that it is more on this side of the spectrum. But is it on that side? How much more? We don't know. Now that's important because when we talk about performance for protein interaction prediction, this is ultimately what the background probability is. Okay? When at some point you will ask, am I predicting better than random? Better than random has to do with what that number is. How many negatives do you pull in? But one thing is clear. The number is not 1. They're clearly, you don't have, the protein does not bind to 10,000 proteins, okay? Not every protein binds to 10,000, so the ratio is not 1. It does not have 10,000 negatives and 10,000 positives. Which means that we have to create a lot of negatives, right? How do we get them? Again, experimentally, the data set from people such as Dimitri is in the 1/10 of that. And so we need 10 more, 100 more, right? 100 more than we have. So experimentally, we clearly don't have them. So you understand what I'm saying, right? So this say is if we had 10k interactions here, this is about 1k. But if the ratio here is at least 1 in 10, so we need at least 10 times this. So we need 100k. Minimally. For this here we would need 100k. For this we would need 1,000k, right? Of negatives in order to have a ratio of 1 in 10 and a ratio of 1 in 100. So where do we get them from? What could you do? Any idea? So I want to train that a protein does not bind to another particular protein. Is there anything we can look at 2 proteins so that we can say, well, most likely they will not bind? Is there anything that you, yeah? No.
The charge of a protein?
So if one protein would be totally positively charged and the other one totally positively charged, that would be one idea, yes. The problem with that is, in fact, that in principle that is a good idea. The downside to that idea is twofold. Number one is that we do not really have for every single protein the charge. So it's only, in order to really know the charge, we need to really in detail know the surface. So this is done by the program from Sharp and Honig, or somebody else in Honig now, GRASP. But GRASP does a very, very good job at compiling the electrostatic positive charge and mapping it, showing it. But really you need the detail of the protein. In fact, even if you build a model, the model may have different surface characteristics, and many models will in fact have different charges. In fact, that's one way in which Berry sort of began predicting differences in functional aspects of homologs, of very, very similar proteins. Now, that's one problem. The other problem is that not most binding proteins really are negative positive. So this idea of a salt bridge, electrostatic interaction, it absolutely works with the protein and the DNA. It does work with protein-protein, but it's sort of more of an exceptional thing. So it would be one thing, but would give you a corner that is very specific, and the system would possibly actually learn something interesting, but not sure that would be the best thing for overall protein-protein interaction. Any other idea? But sort of in that direction is already right, yeah?
Subcellular localization?
One way in which we can look at it is if we have a eukaryotic cell, we have a nucleus, there's an exchange in the simplest way. There's an exchange between extracellular and cytoplasm. There's an exchange between cytoplasm and nuclear, but there's very little exchange between nuclear and extracellular. So when we look at proteins that are nuclear and proteins that are extracellular, we sample them all and we call that the negatives. That's one way of doing it. What's the problem with that? Or what could be the problem with that? In fact, this is what people do. Or it's one way in which people do.
Yeah, the proteins that are extracellular must come from inside the cell, so maybe they interact.
Yeah, but that's what I'm saying. So essentially, the communication is more between cytoplasm and extracellular, but they're most likely not nuclear. So if the cytoplasm and nuclear, and so there's some overlap, but there are in fact very few proteins that are predicted to be nuclear and predicted to be extracellular. That's one way of looking at it. Okay? Yeah?
Maybe the machine learning device cannot learn that two proteins are not interacting when they are in the same subcellular location?
That is an immediate problem, yes. In training the system, we will not be able to distinguish between the nuclear proteins that interact and those that don't, or possibly not, right? That will be could be an immediate problem. The second issue is that it may be that this system actually does not really learn anything about protein interaction, but will ultimately learn localization. Because we give it such a clear negative data set that we'll completely zoom onto that. That is another problem that we could have. What we end up doing is, in fact, this, but the primary data set that we in fact use is anything that nobody has ever observed. We take all the pairs that nobody has ever observed and say, that's the negatives. So we take all the set of experimentally known ABs, and so we have sort of three-pronged approach. We have a high-res A binds B. We have a low-res C binds D. And then E binds not F. So low-res means somebody says that C may bind D in some experiments that we don't trust. But it's good enough for us to say if anybody believes they bind, we will not call them not binding. Okay? So we will remove them. So those we will call negatives here. To stay in the color scheme there. This is negatives. This is positive. And this ultimately will be ignored. Just leave out. Okay? That's the way we do it. But now we get into the question for cross-validation. Okay. These guys here are experimental. These guys are in some sense in silico. In fact, you have to understand, I want to develop a method that predicts that a pair of protein binds that are not known today. So in fact, I will train the network or the device, I will train the device that E and F, the one I want to discover, is not binding. So in training, right? So I will, all the things that I will train are, that I train as not binding, the subset of all the discoveries that I hope the system can do is trained to the system as not binding. Okay? So, which is, you understand what I'm saying? Yes, but it sounds stupid, and it actually is. In a sense, it's not bad because you're not overestimating performance. No matter how bad your cross-validation is, if you train this thing that E and F is not binding, and the device nevertheless learns that it binds, you can trust that there is no overlap. Right? So at least you're totally free of overtraining here, because it does exactly the opposite of what it was trained to do. And if the system can find that out, you're really not worrying about overtraining. So that's the good part about doing something as stupid as that. It is, by all means, it's stupid, but it still is the smart, in some sense, the smartest thing you can do. There's not an easy way to do it better, because if you do the subset localization trick or the grasp kind of trick, you buy into other problems. But now the question that I'm asking is, on my cross-validation, should I do the redundancy reduction only on the positives? Again, the positives are the only experimental data. The negatives are not experimental. But should I do a redundancy reduction on the negatives? Remember, there's this story about case 1, A and B used for training, that I cannot use A for or B ever in any other pairing for testing. Is that also true for the negatives? So the answer is, in the literature, of course not, because it's simulated data, because negatives don't matter, and blah, 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 blah. And of course, reality is the opposite way. So here is 4 different methods to predict from sequence protein-protein interaction pairing, recall, and this is an improvement, so a pseudo-improvement. Everything above here means that negatives are not clustered and it seems to improve, but it's not really improving. The negatives are the same, I'm just reclustering the same data, right? The numbers don't go up. I'm using the same data set, I'm just cutting it, slicing it differently. I'm clustering it differently. And it appears as if not clustering the negatives appears to give it a better performance. In reality, the network zooms into getting the negatives right because it sees correlations that are in fact overlaps. So I have to consider the negatives. And that leads to a situation, and I'm not going to go through this, but where we have to very carefully remove, and from all these possible interactions, we at the end of the day can actually only use a tiny subset. So that is a challenge, because I already start with a small data set, and now I'm redundancy reduction, positives and negatives, and I suddenly have a tiny data set. Okay, that is a big issue. And in fact, I know, so the first issue is, do we have to take all the data that we have, or do we have to reduce further? And that brings us to the question, how much data is needed? How could you find out whether you have enough data for the task that you're trying to solve? What's the simplest way in which you can sort of probe a machine learning device whether you have enough data?
Yeah, you can take out some of your samples and see if it gets worse.
Yes. So
do that gradually until you, at a quick point, it starts to get worse.
So in this gradual curve, so what we somehow expect is, what I'm plotting here is the fraction of the data that I take. Say I begin with 10% of the data, and what I expect somehow is somewhere a saturation. All right? Now, if I saw that for I don't know, 80% of the data, I begin to saturate. That's my, sorry, I have to write this. This is sort of the training performance. So essentially, all I'm saying, I'm not saying that I can generalize. All I'm saying, I can train. Okay, let's begin there. And we could also do the same thing for generalization that will teach us something else. But something similar. But let's begin with the simplest one. Can I train it or not? By the way, why can I not just use the performance? So if my system, by the way, to just make you get used to numbers. If I have secondary structure prediction and my system has 50%, a Q3 of 50%. So a random background is something like 33 if they are equally distributed. Why can I not, from the smallness of the number, immediately make the conclusion that I don't have enough data? I do some machine learning, I have some data, I get this value, and the value is much lower than I expect. Hence, I conclude that I don't have enough data. What's the flaw in that argument? Why do we need to plot this? Ultimately, why do we need this curve? So why can we not just say, well, if the method doesn't work, then I don't have enough data? It's very simple, yeah?
It's possible that it also does not work with more data.
Right, it could be that the reason why it doesn't work is because I didn't put the right input. It could be that I'm just not looking at the right features. If it doesn't work, there are many reasons why it doesn't work. It may not be, and as you correctly say, it may also not work with more data. And that ultimately is the point that we need to see here. We need to distinguish between, is my model not right? Can I not learn it with an SVM? Or can I not learn it with this input? Or from, I have enough data. And ultimately, as long as I see that if we get to my 100% of the data, and we are still in this realm here, then we are still in the part where it's clearly rising, then I know that my data is not enough. Okay, because we're still in the steep increasing region, right? While when we get into the saturation region, close to the 100% of my data, then I begin to see that, well, maybe if I had more data, maybe I could do more. And with more data, I could use more input. So maybe more data would help me. But at least for my current input, for my current way of defining the model, I seem to get with the data into a saturation. So for what I use, the data is not the limiting step. Is the conclusion if I saw something such as this one. If it clearly rises here, then the conclusion is we do not have enough data to tell. Then again, there can be all kinds of reasons. Now, one simple way of doing that is you simply say you double the data, does it improve or not? That's the simplest way, but ultimately Dan is right, this is the best way. Now what we see here, above means that is a pseudo improvement, it's not a real one. And above here, more data seems to be doing good. Down here, more data actually hurts. No, this is a real improvement. No, it's not a pseudo. This is an independent test set for this particular case here. So what we oh, that's for C1. I'm sorry. For C1, more data helps. But for C3, most of these curves are below 0. C3, again, neither A nor B, the class 3 means neither A nor B have been used in training. So if I really want to predict new proteins, more data does not help me, is what this one says. So that means I do know in this particular case through this experiment of the problem is some of this one here. We have absolutely not enough data. Although we have absolutely not enough data, if we had examples like A binds F, and F is completely different from B, so the interaction between A and F and the interaction between A and B is very different. It may even be different domains. I still have to remove that interaction. If I don't, ultimately what the network does, or what the system does, is it zooms into saying A is a protein that interacts quite often. So all it essentially will try to do is sort of predict something like the zip law in a simplified way, in which it says there's some proteins that interact with more, and they essentially interact with everything. Which is a very dumb prediction. It's very difficult to avoid that. Okay, so that's why we have to filter out data and essentially have every interaction only once. And that makes it really complicated to do the cross-validation. So I have to remove important data that I have. I have to do the redundancy reduction and everything on pairs, and I have to do it on negatives and positives. That makes the data set small and makes many things complicated. Now, there are two different ways in which we could do that. So the one way is, I believe I already, did I say that? So one way essentially is the idea that I presented before. So essentially we sample all possible ways in which two parts can bind. The other one is I take these two proteins, put them into the grinder, and essentially look at some overall features between these two proteins. I have in step one a binary decision. So in step one I'm not saying protein A binds protein B here, but I'm simply saying protein A binds B. So I'm putting A and B in the grinder, and I look at some average features of A and B. And for this grinder story, we take again the profile kernel SVM. It's the same thing that we took for the localization. Do you remember that? Okay. Now, the only difference is that in this particular case, instead of having 8,000 input units for three mirror, so 20 times 20 times 28,000 that we used for the subset localization prediction, we now have two proteins, right? So the data set is a little bit more, the input space is a little bit larger. So we have 8,000 times 8,000, which is sort of a considerable number. Those are huge vectors that go into this SVM. Again, that's the profile kernel idea, that we multiply these two vectors. And that brings us to this issue, how many k-mers are really relevant to make the final decision? Can we take the subset localization and sort of predict features such as nuclear localization signals, so how much information is contained in k-mers? And this is for log3. The number is 100,000, 700,000. So this is the distribution of the information content. So most classifications of protein subset localization involve something between, no, but that is not true. This is not for log3 too. This is for protein interaction. Involves something set between 150,000 and 300,000 k-mers. Okay? So it's an immensely large input space. So the information for two proteins to bind is not just in one motif. Okay? Hundred thousands of motifs. They contribute to the information. That's what this curve has here. Okay, but then when you do that, you get a method in dark blue here for C1. So protein A and B have been used for training and testing, but not the pair. And for proteins that have not been used for training and testing, the background here are sort of where we started the best methods that were available. We do not improve substantially. We do improve substantially here for the totally new proteins, not here, because the most important part of the region here is this ROM, and you can see pipe 2. In this part, in this region here, pipe 2 is actually better, and that's the most important region. So we do improve for proteins for which nothing is known, but not quite here. Again, we do improve overall, but again, the most important part is the few proteins that we get right. Now, again, this is based on thousands of patterns, and this is a very tricky machine learning device because you have a huge input space to optimize with a few parameters. Now there are other ways to predict protein interactions. So the first one, I'm going to go through a couple of biological ways to predict interaction. The first one is duplication. So here the simple idea is you go from organism P to organism C, and when you look at all the processes that you have in P and C, so these are essentially all the proteins, and you simply construct a vector in which you look at in P which proteins are there, and in C which proteins are there. And you expect that if there's a correlation between two organisms or between these vectors, then you will see that these somehow reveal something about interactions of proteins, right? So you will see that they are absent in some, so that particular pairs of proteins are, so you cannot do that only on two organisms, now you play that on many organisms and make many pairwise comparisons, and then you begin to see that some proteins are on and off in particular organisms, and you assume that you can infer protein interactions from that. That goes back to work that was initially published by Christoph Zunis and the group of David Eisenberg 20 years ago. Genomic profiles. Now the next idea is the idea of domain copy that is much simpler. So we have a protein A in organism X and a protein B in organism X, and we find an organism Y which we have a protein C that has this domain and that domain, ultimately, or a homolog or something similar, right? So then we assume that in this organism Y these two are merged. They are merged because it's simpler, since they have to interact with one another, it's simpler to put them onto the same gene because you increase the probability that they meet, right? And when you see these cases, then essentially you can infer that we have protein-protein interactions. And that is an idea, again, 20 years ago, was initially published by Terry Gesseland, and simultaneously, almost simultaneously with David Eisenberg. By the way, those are two examples. I find that very interesting in terms of the way science work. Two examples where two groups completely independently of each other invented something that was similar. And in both cases, there's no cheating, absolutely clear that there's no cheating involved. I know the players, I was present, and I somehow was even the go-in-between in some cases. So this is the kind of thing that happens often in science, that you have a new discovery, and they're laying around, and that's the moment when people find it. Yes?
Excuse me, can you go to the last slide? I wanted to ask if we know that protein A binds or interacts with protein D, another protein. Did they try to infer that for protein C?
I'm a little bit confused now. So now we have A interacting with another protein that is not here, a protein D.
Yes.
And now, what is your question?
If they try to infer or see if it works to infer that protein C also interacts
with D.
With D. Given that.
So I do not know the answer to your question. I cannot remember. There must be an answer that I don't know. What I have to tell you though is that both these methods are not able to. So when they work fine, but they cover relatively few cases. So they have relatively few novel discoveries. The discoveries they have are good, and they get better over time because they are driven by more data. The more sequencing, the more organisms you do, the better they get. But they are still suffering from finding few cases. And now you're sort of making another loop. I'm not sure. I do not know. Okay, so the next idea is correlated mutations. We talked about that briefly in passing last semester. So we have a situation of the following type. We have a residue at this position here. What you see is, for simplicity, there's a positively charged residue, not the tryptophan here. Here's a negatively charged residue, not the valine. So now what you may notice is, so these are 1, 2, 3, 4, 5, 6, 7, 8 sequences, or 8 different species typically. And you see that there is a coordinate change of these points. And the idea is that this coordinate change has to do with a constraint in evolution. meaning that essentially we are flipping to another negatively charged residues that has a slightly different size, but you have to maintain the charge. And then if you flip to something that no longer maintains the charge, you really have to maintain some type of the interaction, the V and the W. And ultimately that implies that these two residues are close in space. So the idea is if we find those kind of residues, we can predict contacts inside of proteins. And that in fact is one way in which you can predict contact maps. Here is an example of some of these contact maps. So the gray, I'm no longer sure, the gray is the observation, the red is the prediction for two different proteins. That is work from Debbie Marks. This is actually, is that from the, yes, that is from the transmembrane protein. So that is actually from Thomas Hopf as the first author, the group of Debbie Marks with Chris Under on it. And so these predictions are so good, so these red ones align so well with the gray, the red dots align so well with the gray, that they dared to predict protein structure from sequence for 3 different membrane proteins in that paper in Cell. So you see 3 different structures here. Those are models of proteins for which there is no protein with a similar sequence for which a 3D structure is known. So it's the first time that people in their paper, they had 19 of these. The first time that people really did a 3-dimensional structure prediction of a membrane protein on top without anything that is sort of homology related for very large proteins. And many of these 19, I'm not sure, Thomas at some point told me 5 a few years ago. 5 had been verified experimentally, but clearly that was interesting. And again, all of this is based on the idea that correlated mutations tell you something about interactions, right? Now, since that works on the level of proteins, of contacts in proteins, you can imagine that it also works between proteins. And that, in fact, is the next big method. And again, there are a bunch of breakthrough papers published by that group. I should put that down here. What I put down instead here is the original work from Alfonso Valencia that sort of introduced the idea almost 2 decades ago. The newer methods here is just live off the fact that they have much better data, and that makes a lot of difference, and they use the data slightly differently. That's true too. Here's the paper, eLive, with which they can, for some fraction of the yeast or E. coli interactome here, this tiny fraction they can predict. And the problem is that they need really large alignments. But anyway, so they predict a lot of novel protein pairs. Not sure what is missing here, because my slides are also white. Okay, it's just white spaces. The idea of a mirror tree, this is an idea, that's not the mirror tree. There's essentially, the mirror tree idea is here, you have 2 phylogenetic trees, and you assume that these, or you observe that these 2 phylogenetic trees have a high similarity in the way they are structured, and the resulting distance map are sort of complementary, and from that complementarity and the similarity of the distance, the trees, you infer that the 2 proteins interact. And again, the method works to some extent. Now, the next idea is you have motifs. You simply find in the protein motifs that bind to each other, and when you see these motifs, you say that things bind. Again, there's Andrei Ryshetsky and Hanna Margalit published this almost simultaneously. Again, this is, I believe this is a remarkable story here. We have three simple new ideas how you can predict in a biologically meaningful way protein interactions. And in each one of these cases, of these three here at least, there were two simultaneous publications of groups that are not collaborating and that came on the same, fell onto the same idea from very, very different sides. So here's another extreme example. Here's one example here. This is a genetic, or somebody worked in genetics. This is a protein structural biologist. This is a protein functional biochemist, and this is somebody who is more on the side of computer science. So very different groups coming to a similar solution from very different ends. Now, commonly used, and that is not quite up to date, are all of these. Then is gene fusion. That, in fact, is a signal that is overstated. Domain interaction is essentially the motif that I showed you before. Functional similarity, Go numbers, give an average signal, phylogenetic profiles I talked about. Microarrays, I'll get back to in a moment. So, microarrays allow you to predict protein interaction not using the array data right as it is, but you have to essentially feed the microarray data into machine learning devices, and then you can predict physical interactions. From the array data alone, actually you get associations, not interactions. So if you use it alone and put that into a Bayesian network, then you do actually worse for predicting physical protein interactions. You have to do some additional step, but then you can do it. Here's another idea to use structure. You essentially look at surfaces and interfaces in some sense like the grasp idea that you said, the positively charged surfaces or bits on surfaces that you can use. And in fact, it comes from Chung and Honig. This is exactly the group from GRASP, or Barry Honig is the one behind GRASP. So they look at motifs, learn motifs, structural motifs, and try to predict from these structural motifs that protein bind. That's one of the few bioinformatics papers that was published in Nature. And again, it's sort of here it maps different data sets. That's where they are, their prediction in terms of the true positive rate, false positive rate. And most of these points here, these orange points that they compare themselves to, in fact, are what you would typically call experimental methods. And that is an interesting aspect of protein-protein interactions, that you try to combine any piece of information you can possibly get, and it becomes increasingly difficult to distinguish between whether the result that you have is actually a prediction or an experiment, because almost all methods somehow use experimental information. But that is sort of the state of the art. Back to sequences, we can use the profile kernel, and the question is, can we now sort of go back to oh, no, that part I don't want to do. There was this point about negatives. I just want to bring up this slide again. I showed it before. You see here a map of subcellular localization in 1, 2, 3, 4, 5, and if you call membrane 6 classes. The box is green if this pair of proteins from these compartments interacts more often than expected by chance, is red when it's less often than expected by chance, and it's white if you cannot distinguish. So it's just as expected by chance, a little bit more, a little bit less, but not statistically significant. So what you see is that most of the diagonal is green. So most proteins interact with proteins in the same compartment, and then the problem is if the system zooms into that, you cannot distinguish between those that do and those that don't. But clearly what you see is there's some correlation, but you also see that there are proteins off the diagonal here, or the boxes here off the diagonal that are absolutely green. So some pairs of proteins, for instance, nuclear and cytoplasm. There's a lot of exchange. Again, not very surprising. This extracellular story here is surprising. You would expect extracellular cytoplasm to also be green, but the reason here is that when we compiled this matrix, there was just not enough data for extracellular proteins. But UC2 hybrid has a problem with extracellular. That ultimately is the problem. Anyway, so this is behind the idea of creating the negatives. We use that as a sort of additional filter in the end. Here's a tool from Here's a tool from Christian Dalago. Yes, this is when you do your master in computer science, the way you pick it up. He at least did. So the idea really is that you predict the pair to interact and you plot that onto a grid of the cell. He had a little tool that draws a cell. In fact, here are different upload images that you can upload. These are some that he provides. You can do your own images. And then ultimately the tool tries to sort of get away from what people call the hairball of protein interactions. This is one particular protein interaction in cytoscape where you see the blue protein interacts with all of these gray ones, and the gray ones behind there make this become black. Now, if you have something such as this one, and you are the expert who now has to find something in there, you have, in this particular case, most likely, I can't remember what the interaction number is, but that's the ballpark of 500 or something like that. So now, from 500 points on a screen, very difficult to do. You have to pick out one at a time, or maybe even 1,000. This is very tedious, if possible at all. Mostly what you will do is whatever is next to your eyes, you will take, look at it, and 20 later you will give up, or whatever your patience is, right? So I'm not saying that this is an absolutely stunning image here, but on the same resolution, you clearly see more to actually look at in the next step. So just begin with the following. You see circles, right? There's no doubt. Just see, and here you don't. I mean, you see some in the foreground, but the ones in the foreground here is completely random. Which one come in the foreground? There's no sorting in this mechanism, right? While here, it's not as random. Here it has to do something with where they are, whether they're in the mitochondria, whether they're in the nucleus. There are some, whenever you do your tool, there are some issues. So this is a way to show that this is a nucleus. Some people may find that confusing, but whatever. By the way, when you develop anything visual, try as many people as possible in your environment. Show them and see how they react. Don't ask them whether you understand it. Just observe them while they use their tool and see what they try to, from their eyes and the way their hands work. Try to see what they understand and not. Anyway, so this is a tool that he developed for his master thesis, or one of the things he did for his master thesis, that I hope we will get to a level where we can actually use it.